Stem cells are a class of undifferentiated cells that are able to multiply into any specialized cell type. Stem cells commonly come from two main sources, embryonic and adult stem cells. Due to the stem cell's unique regenerative ability, these cells offer the potential to treat diseases such as heart disease and diabetes. Stem cells have the ability to grow into any cell the organism seems suitable. This is most common with embryonic stem cells when a fetus is in its embryonic stages. The organism starts at a blastocyte, where in the first 3 to 5 days of conception, the embryonic cells multiply into a mass of undifferentiated cells. The cells can then differentiate and become any muscle, organ, or tissue the embryo needs.
There are 4 different techniques used to harvest stem cells: therapeutic cloning, in vitro fertilization, umbilical cord/bone marrow transplant, and induced pluripotent stem cells. Therapeutic cloning is accomplished by inserting somatic cells into eggs, which are then stimulated and begin to divide mitotically, which produce 2 identical daughter cells. A blastocyst, the second stage of fertilization, is then created, where the inner cells are extracted. In vitro fertilization, the eggs are extracted from the donor and placed into a petri dish in the lab under care until the blastocyst stage is then frozen for later use. Umbilical cord blood is collected by cutting away the placenta, where the specialized cells are located and saved for later use. Bone marrow transplant is a procedure to replace damaged or destroyed bone marrow. There are 2 different types of transplant, autologous and allogenic. Autologous transplant procedure uses one's own cells, which are collected from the bloodstream and stored for his or her transplant. Allogenic transplant uses cells from a healthy member, unrelated donor, or umbilical cord blood unit. Finally, induced pluripotent stem cells have been created from an adult cell, which can be found in liver, skin, stomach, etc. This is done through the introduction of genes that reprogram the cell and transform into a cell that has all the characteristics of an embryonic stem cell.
Stem cells have the ability to help us understand how a fertilized egg becomes a complex organism. Medical conditions such as birth defects and cancer are caused by abnormal cell division and differentiation. With a better understanding of molecular and genetic control, learning about these processes can help to discover information about how diseases arise and various strategies for therapy. Stem cells have the ability to replace damaged cells, which is currently being used in treatment of burns and to help restore blood in patients who have leukemia and other blood disorders. Stem cells can also be used to study disease, which can be used to model diseases in a laboratory setting and can be used to better understand what goes wrong.
The ethicality of stem cells is widely debated in our era of science. Even with all the stem cells can offer for medicine, the question of how we obtain these cells is heavily ridiculed. Some argue that at conception, the embryo is a living being and we are taking the life of that being when we are harvesting the blastocyst. It is a delicate balance between those who see the future of stem cell research and those who oppose killing living organisms for science. In the U.S., it is illegal to create, grow, and abort fetuses for research purposes. However, there is a bill that encourages research that would isolate pluripotent or embryonic-like stem cells without the destruction of human embryos.